Thanks to everybody. I'm José González-Pimentel, and I'm going to present the research we are carrying out on how the native microbiome could be useful to control the harmful microbial outbreaks in cultural heritage. I'm talking on the case of an isolated bacteria strain, a species from genus Crossiella, from Altamira Cave. Within the cultural heritage, cave art supposed the first interpretation of the activity of the modern human being representing in paintings and engravings. These remains are well preserved due to the stable environmental conditions in caves, and thus avoiding the deterioration because of adverse climatological conditions. In Europe, Altamira, Lascaux, and many other caves house some of the world's most prominent Paleolithic rock art. However, the massive tours during the past century altered the common condition of caves, favoring the development of foreign and latent microorganisms that colonize the cave walls, threatening the integrity of painting. In Lascaux in the middle of 20th century and Altamira in early 21st century suffered phototrophic microorganisms outbreaks causing severe deterioration of paintings and forcing to close the caves. In addition, several strategies were deployed to eradicate the harmful microbes through the control of environmental perimeters, mechanical removal of colonized sediments, and the use of biocidal, but none of them obtaining the desired results. Furthermore, the use of biocide benzalkonium chloride in Lascaux triggered a fungal outbreak, damaging the paintings. In Altamira, however, only the presence of bacteria was observed without evidence of fungi. The study of microbial diversity resulted in the identification of many species of actinobacteria, such as Actinomadura, Amycobactopsis, Nocardia, Micromonospora, Rhodococcus, Streptomyces, well known for their capacity to produce antifungals, and also one strain affiliated to the genus Crossiella, with no reference on antifungals production in scientific literature. Combination of cultural dependent and molecular biology techniques were used to check the bioactivity of isolated bacteria. They were tested against environmental strains of fungi and pathogenic bacteria such as Escherichia coli, Staphylococcus aureus, Acinetobacter baumannii, and Pseudomonas aeruginosa. Crossiella was able to inhibit the growth of all the different strains of fungi and bacteria used in the assay. The next step was the identification and description of the antifungal produced by Crossiella by means of molecular biology techniques. For this purpose, we use both genomics and transcriptomics approach. In genomics, we extracted the bacterial DNA for sequencing, and the resulting data were assembled for getting the complete genome of bacterium. So once we have the genome, we have the information of all the genes in the bacterium. And thanks to the bioinformatics, the discipline that studied the biological process through computing, we were able to predict up to 47 bioactive compounds produced by Crossiella. Once we got the genome, the use of transcriptomics is essential to identify which genes are involved in the synthesis of the sought antibiotic among those 47 bioactive compounds. Transcriptomics studied the transcription from DNA to RNA, that is to say, the genes or transcript expressed, depending on the condition for the growth of bacterium, to produce different proteins. So we performed several culturing configurations based on the present absence of nutrients to force the transcription of different genes with the goal of identifying the gene or group of genes involved in the synthesis of the fungus. Well, in the end of the research, we will insert the gene in an innocuous commercial bacteria for culturing and getting significant amounts of the high-demanded antifungal, extract the compounds, and subsequent chemical analysis. The chromatogram will be used to characterize the antifungal and finally test the pure compound in a damaged painting or an engraving. As conclusions, massive tours led to the damage of the cave art by the irruption of harmful microorganisms. Traditional strategies carried out to avoid the biodeterioration of paintings in Altamira and Lascaux didn't turn out to be effective. Native microorganisms in cave present mechanisms to synthesize antimicrobials as a way to compete in an oligotrophic environment. Culturing isolation of Crossiella species strain from Altamira Cave as well as the implementation of molecular biology techniques based on genomics, transcriptomics, and metabolomics have shown to be useful tools for new drug discovery, with a potential use in threatening the biodeterioration of cave art.